Good afternoon to you all present over here. On behalf of ISF College of Pharmacy, Moga, Punjab, I, Dr. Saurabh Kose, Professor, Department of Pharmacy, welcomes you all. As you know that ISF College of Pharmacy, under the Aegis of Internal Quality Assurance Cell, is organizing this ISFCP dialogue series. In this view, today we have with us very eminent speaker, Dr. Rajesh Kumar Sharma, sir. I welcome you, sir. Dr. Rajesh Kumar, sir, is a very dynamic academician in the research area of pharmacology. So without taking any time, I would like to read out bio sketch of Dr. Rajesh Kumar Sharma, sir. Dr. Sharma is eminent teacher of pharmacology, toxicology with more than 14 years of experience, including teaching to graduate and postgraduate students. He has also taught and researched in various institutions in India as well as abroad, such as Adigrat University, Ethiopia, East Africa. Currently, Dr. Sharma is working as associate professor in NIMS University, Rajasthan. Dr. Sharma has various publications to his credit, more than 35 years of 35 research and review articles, 3 books, 4 patents granted in the national and international levels. He is a member and reviewer of several national and international reputed journals. Dr. Sharma supervised several research projects for PharmD and MPharm as well as for PhD students in various disciplines and areas of research in pharmacology and clinical research at various preclinical and clinical setups. Dr. Sharma is an external examiner and paper setters for various reputed institutions, including private and public universities. He is appreciated as Best Faculty Award by Dr. B.S. Tomar, who is Chairman and Chancellor NIMS University, Jaipur, for the best performance in teaching and research on Republic Day 2013 function. Dr. Sharma has awarded Young Pharma Personality Award by President of Pharmacy Council of India in National Pharma Summit 2022 New Delhi. His research areas of interest are drug utilization studies, quality of life studies, hepatotoxicity studies, nephrotoxicity studies, diabetic neuropathy, cardioprotective activities, etc. So with this brief bio sketch, now I would like to call Honorable Dr. Rajesh Kumar Sharma to start his presentation. Thank you, sir, and welcome to ISFCP.
Thank you very much, sir. And I would like to be thanks Chairman, sir, and Director ISF College of Pharmacy, and Vice Principal, sir, and Professor Siddharth, sir, and Kose, sir, that given such a good opportunity to give a chance for talk on laboratory animals and husbandry research. And especially thanks to who are joining and participating this series. Thank you very much, sir. I will start. Shall I share the slide, sir, right now? Is visible, sir? Hello, sir. Dr. Kose, sir, is it visible that my slides?
Yes, sir. Slides are visible. Please move it. Next slide, please.
Okay, sir. Okay, thank you very much, sir. So today we will talk about the laboratory animals and husbandry research. Okay, so in pharmacology lab as well as for that other biomedical research. So what are the common laboratory animals are used in biomedical research or pharmacological research? We will discuss about and we will see what are the guidelines are. Guidelines are very important to care of these animals. So as we know, there are a lot of animals we generally use for the various activity, pharmacological activity, or in research. So we can classify these animals who are generally commonly used. So among these animals, rodents are very commonly animals. They are small animals and easily handle or easy to care. Another like mice, rats, guinea pigs, and gabriel and hamsters. Another non-rodent animals are the rabbit, dogs, and cats, monkey, or pigs, etc. Other miscellaneous animals are also nowadays using in various activities, especially in the cancer, like zebra fish. We are going to be used. Even frog. Nowadays, frog are not using because of so many reasons. Uh, Now we come to the individual animal. So mice are very small animals, smallest laboratory animals, very commonly used in all kind of research activity. Okay, reason is that because they are very small as well as they are easy to keep, easily handle, and they require small place to housing, and they breed also uniformly, and commonly strained Swiss albino mice we generally use in various pharmacological activity, or they are very sensitive to small dose of the drugs, very commonly or very importantly used for the knowing of the, it means for the acute toxicity of new drugs or any new entity. They are known species in which it is very possible to grow totipotent embryogenic stem cells in vitro, can form germs lines once re-injected into the developing embryos. Or mice use their tail to help in the thermoregulation. Not only for the mice, even for rat, that their tail is used for them. It is maintain the regulation of the body temperature, and they are also used for the biopsy of the insulin, screening of various activity like analgesic, inflammation, or anti-cancer, or in diabetic also they can be used. They are using the testing of the teratogenicity for the chronic toxicity, especially in the toxicology. And they are very important model for the research in human disease, like I just talked about the anti-cancer, or anti-diabetic, or aging, even the atherosclerosis or immunological disease, or so many other endocrinology study. Next one, we come to the rate. Rate are little bit more, higher animal, or you can say these are little bit larger animal than the mice. They are also commonly used as it is like mice. Okay, they are also commonly used in the various activity, especially used for the different hormone and for the study of the oestrogen cycle, mating behavior, and fertility, as well as for the psychopharmacological activity. They are very commonly used, especially two inbreed original strain we generally use in our common laboratory, like Vista rate as well as SD rates, which is known as the Swango Dowley rates. So just difference only among these animals, just size of their head and tail. Uh, Both, it means all small animals like mice, rat, or guinea pigs, they do not have vomiting center, so they are in that way also they can be used for the uh, oral administration because they do not vomiting. So that's why it's very good, and do not have tonsil and gallbladder, and that's why they diffuse pancreas, so not good a good model for the type 1 diabetes, but they can be used for the type 2 diabetes. Various chemical like streptozotocin or alloxan generally used for the evolution of anti-diabetic activity in another type 2 diabetes. Another, they have the caprophagy, that means they eat their oven stool, and the same like they also regulate the body temperature by that time. They also these rates they can be used like same pharmacological activity, like they can be used for the physiological behavior pharmacology, psychopharmacology, or other activity like cancer or cardiovascular disease, etc. And they can be used for the they generally we can get their isolated tissues like uterus, ileum, and fundus strip for the various activity. Third one is the guinea pigs. These are very docile animals, very small animals like rat or herbivorous. Okay? They eat generally green food and seed and roots. Guinea pigs are not only to synthesize required daily vitamin C, so that's why they are generally given to the vitamins in their water. highly sensitive to stramine. So these mice, which means guinea pigs, they specifically for use for the hypersensitivity activity. Okay, not only for the stramine, for the other anaphylactic sort of activity. Even just I talk about highly sensitive to penicillin and serum contain an enzyme aspergillase which shows anti-leucemic activity. Very susceptible for the tuberculosis and anaphylactic shock, and their same like isolated tissue like ileum and jejunum are used for the biopsy of different kind of the drugs. They are also terminal portion of the ileum used for the just screening of the spasmodic and antispasmodic activity. An ideal model for the enteric amoebasis, hypersensitivity, immune response, anaphylactic shock, and encephalomyelitis, and TB and ascorbic acid metabolism. Other Gabriel, these have like also known as the sand jet or jets. So they have length between rat and mice, intermediate size. They are also easily handled and mild and quiet in nature. They are also using the various pharmacological activity, especially for the anti-epileptic or anti-parasitic or other lipid metabolism or heart disease study. Hamster. These are third most commonly used laboratory animals after the mice and rat, and they are different species according to their geographically, like golden or Syrian hamster and Chinese hamsters. Hamsters are used extensively in oncovirus, especially for the virus, disease, generated disease like influenza virus, respiratory syncytial virus, and vaccine production. And they have also check paws, do not have the lymphatic drainage, and hence they are ideal site for the tissue transplant, such as tumor and graft. European hamsters is very more suitable model for the highly concentrated and prolonged smoke inhalation study. Another Chinese hamster also very commonly used for the cytogenesis. Syrian hamster also used for the some other biomedical research. Armenian hamsters are more specific for the research for the mutagenic activity or other carcinogenic activity because they are very susceptible for studying of the meiosis. Another rabbit, very commonly used for especially for the diabetes or biopsy of the insulin, very docile animals, or mainly used for the parenteral product, to, especially for the pyrogen testing of the parenteral product in all pharmaceutical industry, or can be used for the heterogenization of some other drugs testing in eyes that may cause the meiosis and mydrosis and used for the large volume of parenteral for the screening and biopsy of the insulin, diabetes, or curing mimetic drugs. We commonly use in our laboratory New Zealand white rabbits, okay, that is specific for the evolution of anti-diabetic drugs, diphtheria, TB drugs, cancer, and heart disease drugs. They are especially used for the screening of anti-fertility drugs and therapeutic study. And their skin also very sensitive or may cause the irritations. And good model for the production of antibodies and anti-serums. Even instead of the intact animal, some isolated tissues can be used for the preparation. Isolated preparation can be used like heart, isolated heart and duodenum and ileum for the various drug activity, and can be used for the cosmetic and pharmaceutical products. Uh, it can be used for the cardiac connective tissues, free of the connective tissues, even as an animal of the choice for the many cardiac studies, or sensitive to the stimine. They also do not have the organ center. And can be used for the pharmacokinetic and pharmacodynamic study of the drug. Or enzyme atropine esterase present in the blood, which trigger the atropines, but commonly used for the uh, topically in eye. Monkey. After that, small animals, if drugs are effective, so to determining of their efficacy and safety, we can go for the monkey, large animals, because they have similar size and functionally like to man, suitable for the undertaking psychopharmacological activity and their uterus also similar like human beings, that because the regular menstrual periods. So they are commonly used for the pharmacokinetic study, or very useful for the CVS or CNS or GI and fertility drugs. Require regular checkup for the rabies, TB, and timely immunizations. Used as a primate model to study drug metabolism. Dogs also used especially for the cardiovascular drugs. Most prefer large experimental animals comparative with the even after monkey. Used for the studying various antiarrhythmic, cardiovascular, and autonomic drugs. The advantage of the dogs because they are small, alimentary tract, and easily get trained. Mongrel and beagles, that's dogs are very commonly used because they are suitable to work on that these animals and size, moderate length of the hair, coat, etc. Stomach and intestinal tracts similar like the human beings. So very good model for the antidiabetic activity, for the reproduction, for IBD like ulcerative colitis, and open heart surgery and organ transplantation, central nerve systems, safety pharmacology as well as for the toxicology study. It also can be used for the especially for the cardiac drug, especially for like digitalis or other drugs. And it can be used for the transmission of the vitamins and minerals to the fetus and newborn. They are also used for the pharmacology and toxicology, and for the oncology and chromosomal abnormality study. Another one is the pigs. They generally use for the for production of the conventional insulin, and they are hairless, so they can be used for some drug effect on the skin, and susceptible to bronchitis and pneumonia. So some other antibiotics that you can use. Alimentary tract similar like the human, and especially important model for the cardiovascular as well as atherosclerosis and MI model. Another zebra fish can be used especially for the anti-cancer drugs, and show the because they are developing stage embryogenic some drugs those affect the embryogenesis. In that way, we can use for some anti-cancer drug or other anti-fertility drugs. So they are suitable model for the studying vertebrates, embryo development, and genetic techniques. Different stages of the development can be observed under the microscope. Frog, which we are not using for, but they can be generally used to, and different in pharmacology laboratory. Oxygen can pass through their highly permeable skin and split largely through their skin. And camouflage is a commonly defensive mechanism in the frog. And anatomically, their heart contains three chambers, so which is different from the other member used in the experiment. Chicken can be used. They are widely used because of easily availability. Good model in the toxicology and pharmacology study. Only the model for the study avian disease. And a very important model to develop in the chicken are the chick comb method, chicken blood pressure measurement, heart rate, and EEG, etc. Pigeon also very good screening model for the antiemetics. They also used for the standardize of the cardiac glycoside like digitalis. We know about the noding model, biopsy of the prolactin, and used for the screening of intravenous anesthetic drugs. So instead of these animals, we can commonly use like other genetically modified animals like mice or rats, especially for the specific activity. Now we come to know the what are the CPCSA guideline for care and safety for the animal facility. CPCSA, that is the committee for the purpose of control and supervision on experiment on animal, that is the name, full name. But nowadays, last before 2 months, that name is changed. Only the name is the CCSCA. That is the site. Here the government changed the name CCSCA. That is the CCSCA means that committee for the purpose and supervision of the experiment on animals. So that guidelines, what are the goal of this guideline, and what are the regulation and rule and regulations are there for the caring of the animals is very important, especially for the experimental animals. So just we know about, because this is very important or very necessary to care of the animals even before, during, and after that performance of the experiment. Some other guidelines are there. Breeding and experiments on animal like control and supervision rules came 1998 after the amendment in 2001 as well as in 2006. So this CCSCA, that this under the control of the good laboratory practice, introduced the grade of the 3R principles, that is the reduction, refining, or another is the replacement. After that, another fourth are also included, that the responsibility of uh, researcher, that who is going to investigate the effect of the drug in animals. So this CPCSC under the government of India, they compose the committee member of scientific community, regulatory authority, animal activists, or at least 10 experts are there, 3 other official members, generally including the CPCSC committee. So, just we talk, that is, it comes under the Ministry of Environment and Forest, Animal Welfare or Animal Husbandry Department. It constitutes the IAEC in institutional level or IBSC, especially where the hazardous chemicals or hazardous drugs are experimented on the animals. So IAEC, that is Institutional Animal Ethical Committee, or IBSC, that is Institutional Biosafety Ethical Committee. So, if suppose, because this IAC constitute only for the small animals, or for the large animals, make it the recommendation to the SCLA, that is the subcommittee on the large animals. Here, IAC composition by the institution. Here, biological scientists, one biological scientist will be there, 2 scientists from the different biodisciplines, whether it's the biotechnology or other, department like zoology. One veterinarian involved in the care of animals, a very important part of see the protocol and whatever the changes required is must be included. And one scientist in charge of the animal house facility, regarded all kind of the facility by food, drainage, or whatever the facilities we will discuss next. And chairman generally the head of department of pharmacology as well as the institute of head of the institute, considered among any about 5 members. Others member main nominee, who is main nominee and link nominee. Another scientist from the outside and socially aware nominee nominated by the CPCSC. So main goal of the CPCA for the safety for the animals. So our target, that CPCA target to protect the animals any kind of the cruelty. So just we talk about the main target or main goal of the CPCSCA to guideline is promote the humane care of animals using the biomedical as well as in other research area, and to avoid any unnecessary pain or any kind of the stress before, during, and after the experimentation in this research. They provide the all kind of the guidelines for the housing, what kind of the housing should be there, how to take care of the animals, what are the breeding facility and maintenance of other necessary things are required, and source of the experimental animals from where generally we procure the animals were authentic or uh, breeders, or acceptable experimental procedure for the anesthesia and euthanasia, because it's very important anesthesia, because during the research animals should not be stressed by any kind of experimentation. So anesthesia should be given, and after that completion of the anesthesia, completion of the experiment, that euthanasia, that means the painless killing is very important to reduce the stress. So GLP, that it is just good laboratory practice to assure the quality, maintenance, and safety of the animals. Actually, that enhance the animal well-being, quality in the pursuit of advancement of the biological knowledge that is relevant to the human and animals. So just we were talking about the veterinary cares. So a veterinary is a specialist, or we can say it is he is a very known person to how to handle the animal, how to care the animals. So a full-time veterinarian should be in the large animal house, or where the large animals are kept, or for it may be considered for the part-time or consultative if the small animal house is there in institutional level. He daily observed by animal housekeepers and veterinarians that the observation should be there. The duties of veterinarians are the establishment or appropriate policy and procedure in animal house because he's expert, or he can review the protocol and proposal according the need. Animal husbandry and animal welfare is his duty, and he monitor the occupational health hazards contaminant if any kind of chemicals are using during the experimentations, and if any contagious disease or any infectious disease is there, then he should control, like zoonosis control program he carried out, and supervise animal nutrition and sanitizations. So how from where we generally animal procure? Always we procure generally. If suppose any breeding facility is there in our animal house, then no need to be procured from outside. But if suppose we only have the animals, small animals, we do not have breeding facility. We have to procure from the outside, the authentic breeders. So there should be lawfully from where we are purchasing or procure. Each consignment of animals should be inspected for the compliance, and procurement specification is there for what are the reason why, how many animals are used, what are the facility should be used to caring of animal or procurement or transportation. Everything should be mentioned. Even we have to be submit our protocol where we are going to be procure the animals. So if animal we are purchasing or procuring, that should be quarantine, should be keep separately in the animal house before keeping in common animals. Already those are stuck. We have. If suppose we can get the animal from the outside, especially for the transgenic animals, we can get the license from the Director General of the Foreign Trade. So these are the requirement for the transport of the laboratory animal by the road, rail, and air. So that what type of case material should be there, how many animals we can be keep in the one case, what kind of the distance should be there in all animals. They should be comfortable. They should not be stressed during the transportation. So these are the criteria. And after procurement animal from the from the outside, that quarantine should be there. They should be keeping separately to prevent any microbial or any infectious disease. It minimizes the chance for the introduction of the pathogens into the established colony. For small animals, we generally keep animals for the 1 week at least quarantine in separate room. Or for large animals, we generally keep 6 weeks for the large animals. Or if newly received animals should be given a period of the physiological, psychological, and nutritional establishment before their use after during the quarantines. Okay, so that is the very important. And if it is any kind of the disease is there, that we should keep us separately, completely from their colony, or we can give the immunization or vaccination for animals. Eliminate anxiety and possible psychological and behavioral change due to species conflict. And another important thing that is separation room should be there for the different species. Not like that if you are keeping the mice in the red room, or reds are keeping in guinea pigs. So all separate species should be kept in the separate rooms. So these are the individual ventilated cases. For the non-human primates, that is, they need to be at least 6 weeks for the quarantine separation to prevent any kind of the zoonosis. After surveillance is very important. that should be observed for the sign of the illness, injury, or abnormal behavior by animal house staff. And with the care, that is the person is caring the animal that should be very trained, experts in caring of many animals. If even the caretaker should take the training from specialist for caring. Animal that show sign of any contagious disease should be isolated from the healthy animals in the colony. Or if an entire group of the animals known as the believed to be exposed to an infectious disease, that group should be kept separate. That should be in the treatment or prevent any kind of further infections. So these are the some diagnostic and treatment. Generally, these are the biochemical or hematological data of the individual. We can say CBC parameters of individual animals, and by CBC and LFT, liver function test or some uh, kidney function test are there separately. For preventing medicine program, vaccination, ecto and endoparasitic treatment should be given, especially for the transit in transgenic animals. These are the hairless animals or mutant animals. They need special care in animal house, separate because they are very susceptible to the disease or infections. So protection is especially assured for their health. System to prevent spread of the disease may include facility design features, contaminant isolation equipment, and use of the SOP. SOP is very important to keep the animal care. Training of animal care research staff is essential. Time to time, their medical checkup is required for all animal take care, even the staff of animal house. Cell tissues, fluid, and transplantable tumor that are to be used in the animal should be monitored for the infectious or parasitic agent that may cause the disease in another animals. So these are the laboratory services like necropsy, histopathology study, microbiological, any kind of the contagious disease, any kind of the pathological parameters, serology, or some other parasitology or other specialized tests. Education for the technical person, for whatever that person who is caring all the time, he should be trained. That personal hygiene of the person is very important for the not only for the caretaker, even for the researcher, because all staffs those are maintaining uh, personal cleanliness. We should wear the PP shower, take shower including the entry of the animals, change of uniform and footwears. Whatever the footwears and inside uniforms are using that should not be bring outside. Okay. And use disposable gloves and mask, head covers, coat and coverall and shoes covers. And if refusable to be decontaminated by any authorized laundry services, if it is outside is there, then it should be separately given to laundry service. Some experiment with the hazardous agents, especially just when you talk about the hazards chemicals using during the experimentation. So here separate hazards biosafety hazards committee is there. So he that committee should monitor the all kind of experimentation during the hazards agent. Location of animal facility that should be completely free from it means any kind of the sensitive use. Animal house shall be far from any human habitations, not exposed to dust, smoke, noise, because noise-free area should be there. Because animal otherwise they may affect their health. Wild rodents should not become insect and birds. Experimental areas shall be separated from the housing or laboratory animal facility. Suppose laboratory animal house is there, only 50 to 60% inside constructed area should be there for the only animal rooms. At least 40% area should be free from the rooms and etc. Like that should be proper corridor to movement of person etc. For other services, 40 to 50% such as stores, washing, office and staff, nursing room and quarantine and controls are there. Other physical facility control over vermis, it is pest control and contamination associated with the housing of animals, delivery of the food, water, bedding. what kind of the food is given to the animals. It should be pellets, should be free from any kind of the contamination. Even water should be given the RO water, not given outside tap water. Maybe chance of any heavy metal or other chemical. Bedding should be has whatever we are using the has that should be dry or should not be. Uh, it means that should be also free from any contamination. Wall and floor should be sturdy. That should be sturdy means that should be easily cleanable and durable for the proper disinfectant treatment should be there. Adequate contingency plan to cover such emergency as a flooding and fire or the breakdown of the lighting, heating, cooling or ventilations or entry to authorized person only the enter or disease prevention and general animal welfare should be there. So these are the physical facility should be there like proper what kind of the building material that should not be absorbed by any kind of uh, chemicals. It should be very uh, uh, free from any kind of contamination. Corridor should be proper, that should be movement. All kind of the utility like water, etc. Or other drainage system should be there. Animal room doors that should be very durable or strong. Okay. Exterior window, especially for the large animal, windows is required, but small animals I don't think so. It is required for the exterior window. Floor should be cleanable or drainage system should be proper. Wall and ceiling should be proper. Storage area should be separate from the animal house, like maybe it is from the main animal rooms. Other facilities should be proper. Environment, especially if you talk about the environment like humidity and temperature should be maintained up to the 18 to 29 degrees Celsius. For the larger animals, they can be used, we use 7 degrees Celsius. So thermometer should be there. So that's why we, install the air conditioning to maintain the temperature. Humidity that should be 30 to 70%. Heating, ventilation, especially during the winter season, proper heating should be there. Ventilation, AC system should be designed with the 12 to 15 air cycle hours. And fluorescent light less than 400 lux, that is uh, generally measured by the lux meter. That should be especially for the rodent facility. Time control lighting system to maintain the regular diurnal lighting cycle. Emergency power should be made available in the event of the power failure. Some animal husbandry housing system need to facilitate animal well-being, meet research requirement, and minimizing the experimental variable. Housing system should be properly adequate space, permit freedom of the movement. Whatever we discuss about the content requirements, okay, comfortable environment, escape of the proof enclosure that confines animal safety, easy access to food and water, and adequate ventilation, meet all biological needs like temperature, urination, defecation, and reproduction. If suppose breeding license is there, then we need reproduction. Keep the animal dry and clean consistency with the species requirement. So these are the size of common, especially for the rat and mice, um, these cages. And instead of this animal care, we need to be maintain the records and we have to be work according to the SOP. So in record keeping, animal house staff record for technical as well as non-technical person, health record of the staff and animals, all SOP relevant to the experimentation, care and breeding and management of the animals, breeding stock, purchase, sale records. It means all kind of the records we have to be necessary. And IAC minutes, record of the experiment conducted with the number of animal use that is required the form D in the CPCC according that guideline. Mortality postmortem record wherever required, especially those animals after the sacrification, how we are going to be decompose these animals. Clinical record of the sick animals, training record of the staff involved in the animal activity, water, feed, whatever the things we have to be keep in a record form. SOP is required for the animal husbandry, animal maintenance, for the breeding, for animal house activity, microbial testing, experimentation. So for individual SOPs or record maintenance is very important or necessary to care. These are the contents of the SOP. They should be keep the name of the author or title of the SOP, date of the approval, reference of the previous SOP with the number and date, location and distribution, and what are the objectives. Detailed information regarding of the instrumentation, what kind of the instruments are going to be used, number of the name of the manufacturer of the reagents, if suppose any chemicals are using, normal value of the all parameter, histopathological and biochemical parameters, some others identification of risk assessment also we need. Anesthesia, that is anesthesia generally when during the experimentation, painless procedure should be there. Okay, so we should not be directly, uh, you know, with the pain procedure should not be used, so we have to be use some pre-anesthetic medication or anesthesia. So these are ketamine, pentobarbitones, and thiopentones, and urethane we are generally used. These are the dose according the animals. Euthanasia is the procedure where the painless killing without any stress or any uh, by using of any chemical. So it is performed by proper procedure. So the method should be cause death without causing anxiety, pain or distress with a minimum time they face, minimum psychological and physiological disturbance. That compatibility with the purpose of the study to minimum emotional effect on the operators. And location should be separate from the other animals, otherwise they may suffer with any kind of other stress. Tranquilizer must be administered. So these are so many physical method and other inhalation of gases are used, drug administration. Nowadays we commonly use the ketamines, overdose with the ketamines or other dislocation, electrocution methods are used. Nowadays some physical methods they are not recommended. Only the need of the depending on the requirement we can use this method. Some laboratory animal ethics are there. All scientists working with the laboratory animals must have deep ethical consideration for the animals. So even we just already talk about the SOPs, very important. These are the regulatory aspects. Some this is firstly, you know, Act or you can say regulation that is the prevention of the cruelty to animal PC Act came 1960. After that, CPCSCA it is formulated that is same the things in the PC Act which is converted to CPCSCA in 1998. Then already we know that IAC under comes in the CPCSCA. Some guidelines are there. Guideline for the laboratory animal facility in 2015. Guideline for the reuse and re-radicalization of the dogs. Guideline for the reconstitution of IAC and guideline for the preparation of the SOPs, care and management of the equine used in the production of the biologicals, and some guideline on the regulation of the scientific experiment on animals. So these are the quality of laboratory animals. What kind of uh, animal house should be there? When we enter or exit the animal house, that should be separate for the individual uh, species or strain. Suppose if you are entering the rodent, then we have to be entering the rodent side or exit in a separate. Or if you are entering in the another rabbit or pig facility, we cannot be entering the another uh, side where other rodent or other facilities are there. So here entry and exit register is there for the visitors. They should be mentioned if suppose animal they are carrying from animal house to the laboratory. They should be mentioned everything. These the vivarium anti-air curtains or these separate shoes racks and some separate shoes racks are separately waiting. So motivate with the 1% germicide up to the protectomate to prevent any kind of the dirt or dust or contamination. So before entering of animal house, hand wash before entering, air shower is required. So air shower is there. Then overall cabinet with the UV light and a complete overall, you can say PP kit is need to enter in the animal house. Is that is the clean corridor to proper movement or to carrying of their anything. Emergency exit or autoclave linkage where generally whatever we are using inside the animals, especially cage or other instrument that should be uh, sterilized by the autoclave. So these are the monthly activity schedule according what to the analysis of the water, food, feed, bedding, and other IVC monitoring, etc. Other material whatever we are using, IVC system. This IVC ventilator. So these are exhaust supply and enrichment for the animals, especially for suppose for the well-being or for happiness for the animals. We can add some extra things. For example, in dogs, hanging toys do for the dogs doing the exercise, social contacts during the house in the cage. For the rodent, corn for bedding material for the nestling and during the opportunity, paper cutting for the pregnant babies, etc. Light intensity between the 130 to 325 lights. Okay, even we can add some music system in the animal house to best enrichment. Even no smoking or pollution around the animal house area. Should not be smoke, should not be eat, should not be drink any kind of the things that should not be taken in the animal house. It should be completely free from uh, any kind of smoke or other things. So just these are the some 10 animals say it's most dangerous species in the world, but it found it's like this one. And for the biological waste collection, there are three color container is there. Black is the discarded medicines. Yellow is the anatomical waste, any kind of the tissues or soil dressing or plastic and soil items. Or green, blue it should be there, chickens or needle or any sharp material. For the dog facility entry procedures, just already you know about the should be cover and make entry the register whatever that here and there. And centrally air conditioned for the dog facility. Or these are some protection from any generally like beta we talk about in India, some to prevent the inequality. So these are the some organization against animal testing. Suppose we talk about the PETA. So this is people for the ethical treatment of animals, which is in India. This is organization to prevent any cruelty or some others like BUAE, British Union for the abolition of the sections. So they based on the depending on the different country, there are different organizations are there. These all organizations came after that series of various community examples. So here some example of the silent drug discovery that involve the use of animals like diphtheria, insulin, controversy. So they can general use and these scientists they were they discovered these drugs. So even suppose animal we are using the experimental during the research. If it is necessary, then only we can use. Otherwise, we can use some alternative or substitution of these animals. Okay? So need to alternative to animal testings, in vitro model for the testings, recent advanced stem cells drug discovery we can use. So our target or CPCS is that is 4 R we have to be used. Replacement means if it is non-animal methods we are going to be used, that should be minimized, replace the model. Or reduction if the researchers obtain comparable level of the information from very few animals, we can obtain more information, the more animals. Refinement, that means whatever the more methods we are using, that should be stressfulness, minimum stress, minimum pain, and should not be that should be free from any kind of the hazards. So these things we know how we can reduce the alternative animals, how we can refine the refinement of the alternative by using suppose just we are talking about instead of the larger needle we can use the small needles, use non-invasive techniques like MRI, ultrasound, use other less sensitive species, use smaller dose, test can be added at the earlier feasible time, improve housing condition, etc. And we can replace the animal by using of the other alternative models or we can substitution of the other models. And last one is the responsibility. That means responsibility for the researcher is very important. It should be very reflect their integrity, honesty, and scientific correctness, appropriate and reasonable use of the level programs. These are the alternative models. In vitro models comprise patient-drug database, stem cell genetic testing models, non-invasive MRI, or another microbiology models we can use. These are the regulation in India, based on law and regulations are there according to the government regulated in the particular years. And our conclusion, this so that is we have to be with the minimize to animals, but it is not completely we cannot divide the animals in a study because we we can use other substitutions, but 100% it is not possible like the animals. So we have to be animals, but with the minimum quantity or other things. So animal testing is integral to the safe for human health because it is need, it is compulsory or necessary for the preclinical study. Otherwise, without preclinical study, how we can go for directly clinical research? So it is necessary or it is essential part of research in biomedical research. But other side, we can see abolish the animal experiment or some animal right activists are there, existing authorities are there, scientific authorities are there. So we have to balance between the both research or other things. So thank you very much. Uh, this presentation. Thank you very much, sir.
Thank you, Rajesh sir, for your an insightful talk on laboratory and husbandry research in the pharmaceutical sciences. I'm definitely sure that this talk is going to be very useful for preclinical and clinical pharmaceutical research scientists. You have touched upon various aspects of preclinical laboratory animal handling, their storage and their maintenance. And the slides were wonderful, containing all the pictograms which are the need of the hour. So, on behalf of ISF College of Pharmacy and Internal Quality Assurance Cell, I am thankful to you, sir. Once again, you have taken your really really wonderful time from your really busy schedule on a very short notice. So, on behalf of ISF College of Pharmacy, I am one again thankful to you for
Thank
accepting
you.
our
Thank you.
invitation and delivering such a nice talk, sir.
Thank you,
Thank you
sir, for
once again.
giving such a good platform for providing talk about the little research. Thank you very much, sir.
Thank you, sir. Thank you so much.